Okay, and thank you for the opportunity to present today. So I am a PhD candidate at McMaster University in Hamilton, Ontario, Canada. And today I will be talking to you about identifying anti-pneumococcal molecules in the upper respiratory tract microbiome. So I work in labs supervised by Dr. Mike Surratt and Dr. Don Bowdish, and we are interested in studying respiratory tract diseases such as pneumonia and the mechanisms that play a role in inhibiting pathogenic bacteria like Streptococcus pneumoniae. So, pneumonia is a serious respiratory infection characterized by fluid buildup and other substances, such as bacteria or fungi that accumulate in the air sacs of the lungs. And according to the World Health Organization, pneumonia is responsible for over 2 million deaths each year, and is one of the largest causes of death globally due to infection. So this accumulation of fluids in the lungs can be caused by the inflammatory immune response to a bacterial infection. And some bacteria are really good at attaching to our epithelial cells in the airways, where they can replicate and make their way down to the lungs. Streptococcus pneumoniae is one of the most commonly identified causative agent of pneumonia and can enter the airways through inhalation of respiratory droplets. So pathogens can get into the lower airways by first getting into and then colonizing the upper respiratory tract, which is the gateway to lower respiratory tract infection. So this is the pre-infection stage where the bacteria adheres to the host cells and replicates and has the opportunity to evade host immunity. And this colonization is of concern because it can lead to the spread of the pathogen to other parts of the body, other than the lungs, such as the blood, the ear, the brain, and causing diseases like meningitis and so on. So by inhibiting the colonization of Streptococcus pneumoniae, initially we can prevent a variety of these diseases before they reach the point of infection. So if we look at who else is present in the upper respiratory tract, we find resident or commensal bacteria, which are the bacteria that cohabit within the host. So these commensals act as guards or a first line of defense that inhibit pathogenic bacteria from colonizing. And they do this through contact-dependent interactions. So, for example, by injecting a toxin directly into a target bacterial cell, or contact-independent interactions like competing for nutrients or releasing antimicrobial substances into the surrounding environment and not allowing the pathogen to thrive. So I will specifically be discussing contact-independent interactions. So these include antimicrobial peptides or AMPs and other secreted molecules. And rather than looking into other environments to find bacteria that have these antibiotic properties, we want to investigate the upper respiratory tract environment because these bacteria and their products are much less likely to be toxic to the human host and have possibly evolved mechanisms to defend against harmful respiratory colonization. So, again, commensals have evolved ways to interact with the host to prevent colonization by invasive pathogens. They can directly inhibit the growth of pathogens by producing antimicrobial factors and by competing for nutrients and adhesion sites. But the other role of these commensals is to interact with and prime the immune system by stimulating the host cells to drive expression of antimicrobial factors. So in combination, all these mechanisms allow for the colonization resistance of pathogens and assist the host in preventing infection. So a healthy and stable microbiome is protective, but when there is a disruption in the microbiome, so for example, during the overuse of antibiotics, it can lead to the microbiota becoming less diverse and leading to decreased colonization resistance. So here we see that there's uncontrolled growth of the pathogen, and this high bacterial load in the upper respiratory tract can lead to the bacteria entering the lower airways, or the pathogen might attach to the epithelial cell and disrupt the epithelial barrier and enter the bloodstream, where it can lead to systemic infection. And in some frail individuals, both the immune and microbial structure can decline, leaving these older adults more susceptible to respiratory diseases such as pneumonia because of the decreased colonization resistance. So if we look at the airway microbiome profile of healthy individuals, we can see that there are some dominant groups that are present in healthy adults, such as Staphylococcus, Propionibacterium, and Corynebacterium, that are not as prevalent in the frail elderly population. And it looks like they're instead being replaced by Streptococcus groups. So these commensal bacteria that are thought to be protective are lost as we age, and it looks like the microbiome shifts from these dominant groups to other dominant organisms like Streps and Prevotellus. So what we know so far is that the colonization of Streptococcus pneumoniae in the upper airways can lead to bacterial pneumonia. And we know that commensals in the upper respiratory tract can protect against this colonization. And we have profiles of healthy individuals that can help us predict which groups are associated with this protection, but a lot of the mechanisms are still unknown. So we believe that there are multiple mechanisms that prevent Streptococcus pneumoniae colonization that are yet to be identified and described, and this is what we're aiming to do. So the first goal is to isolate diverse bacterial strains from the human upper airways and to identify the ability of these isolates to inhibit Streptococcus pneumoniae. And of course, look for possible mechanisms of inhibition using biochemical and computational approaches. So the microbiota varies between individuals. So we created this diverse strain collection. We collected samples from 21 healthy individuals. These were nasal swab samples from the mid-turbinate nasal cavity. And these samples were cultured on a whole bunch of different media types to capture the diversity of the respiratory tract. And what we see is that there is a large variation within our samples. We isolated over 2,000 strains representing 21 genera. So to determine the microbes with antimicrobial activity, we screened against Streptococcus pneumoniae and looked for zones of inhibition like this one. We identified 46 isolates with this activity, representing 7 genera and 13 species. And this inhibitory activity has been observed to be strain rather than species specific. So here I have highlighted one of my inhibitory strains, classified as Micrococcus luteus. And again, I will mention that in many cases, not all strains of the same species produce inhibitory molecules. So this is exactly the scenario here. In the photo, I have 8 Micrococcus luteus strains. They're all the same species. And we can only see a zone present around one of the strains. I will refer to this strain as GC 1842, and we were interested in finding whether the strain can inhibit other species other than Streptococcus pneumoniae, and we found that it was able to inhibit other streps, but not all. So it can inhibit Strep oralis, Strep mites, which are very phylogenetically similar to Strep pneumo, but we don't see the same activity on lawns of other streps like Strep sanguinus and Strep gordoni. So it turns out that it is actually pretty narrow in terms of the range of species that it can inhibit. So to identify the bioactive molecules that are causing this inhibition, I'm using 2 approaches: activity-guided purification and computational comparative genomics. And this here is a summary of my method. So I first grow the bacteria, allow it to secrete its compounds into the agar, and to purify these compounds, I use a number of solvents of different polarities, as well as liquid chromatography techniques, which allow me to separate the molecules and then assay them for activity. And then finally, we use mass spec to identify the mystery molecule. So we ended up successfully purifying a molecule, and we do have an accurate mass. We still need to do some work to get more details on its structure, but what we know so far is that there are other peptides of the same mass. So brettanomycin, geninthiacin, which are both isolated from Streptomyces, and neobrettanomycin, which is found in M. luteus, which is the same bacteria that I am working with. Next up is NMR. So this is used to predict molecular structure. So far we have proton NMR data, but we still need to do other types of NMR to get a better idea of the structure. And another angle to find potential antibiotic molecules is through genomic analysis. So we have sequenced and assembled these strains, or the genomes of these strains. And we know that biosynthetic gene clusters can produce antimicrobials. So here are two gene clusters, which are groups of genes that are predicted in the active M. luteus strain that are not found in the non-active strains. Both of these are predicted to code for a thiopeptide, which is a class of bacterial-produced antibiotics. And interestingly, these clusters have not been previously seen. So we might be looking at a novel antimicrobial. So in conclusion, we've identified a number of upper respiratory bacteria that have anti-strep pneumo activity, and we see a lot of diversity in terms of the patterns of expression and strain specificity, which suggests multiple mechanisms in which these organisms inhibit pathogens. The focus has been on the molecule of interest from M. luteus, where we are now looking to find its structure. And as for next steps, we want to find if more of these active secreted molecules can be found through activity-guided purification and comparative genomics. And ultimately, the goal is to find if these will be toxic or effective against host cells. So we will evaluate these in a more relevant model than an agar plate, such as cell or tissue cultures, and then move to preclinical mouse models. And I just want to point out again that this presentation was focused on M. luteus, but I do want to highlight the strain diversity again in another group. So this is a phylogenetic tree of the Corynebacterium strains that we have in our lab, where again we see how the different strains behave differently. So the colored circles here represent activity against Streptococcus pneumoniae in different conditions. Those marked with red circles inhibit strep pneumo when they're grown on blood prior to methanol extraction, and those marked with a yellow circle are strains that are just grown in our regular agar without blood. So again, there's a lot of variability in terms of the activities within species, which I think is really interesting. So I'd like to thank my labs, of course, both the Surratt lab and the Bowdish lab, the lab members for their support, and Doctors Surratt, Dr. Bowdish, Dr. Hirota, all at McMaster University, and the other individuals I've listed. So thank you for your attention, and I'd be happy to answer any questions.
Thank you for that wonderful presentation, Nancy. It sounds like you have some very exciting future directions. There's a question in the chat now. It says, have you conducted a comparative analysis of the effectiveness of non-specific protection factors of the upper respiratory tract and microbiota? Which of these is more important in antimicrobial protection of upper respiratory tract?
Okay, let's see. Phagocytitis, okay. Yeah. Okay, that's an interesting point. So we're actually looking into the environment and the step right before this. So before the point where the microbes are getting into the epithelial barrier and into our like past that barrier, to prevent this type of like this immune response. So we're looking into the interactions to prevent colonization. prior to having this like overgrowth of bacteria. And so we won't get to the point of phagocytosis and so on.
Thank you. And I believe Dr. Fish has a question as well.
Yeah, great, great presentation. Exciting data here. So, what you commented is that sort of many different strains might be contributing to this protection that you're seeing, and it's all happening in the upper respiratory tract. I wondered, have you looked at whether it's what's the contribution of sex? What's the contribution? I know you're only focusing in one region, but do you think in different parts of the world you're going to have different, type of colonization that would also influence protection? So sex and whether there are any epigenetic contributions based on, environment.
Yeah, definitely. So the samples that we have, we tried to make these as variable as we could. These were sampled during the COVID time, so it was a little bit hard to get these samples, but we aimed for a diverse age range. So these were individuals between 20 to 80 years of age, and different sex. And even within this small group, we do see a lot of variation. We have done a lot of other like 16S profiles previously with bigger groups, and other studies have also shown that yes, region does have an impact. Age, gender does have an impact. Really what we're looking into is not only age, but age in terms of frailty. So we could have older individuals who have a microbiome, an upper microbiome that looks more similar to younger, and that would be considered healthy. But what we are trying to see is what happens when they reach this point of frailty where coming across any microbe can have this like dramatic shift in their profile.
And if you had to hang your hat on a really important microbe, which, let's say in the frail, because those are the ones who are most susceptible, which one would you say it's really important to focus on to analyze those bioactive molecules?
Yeah, that's an interesting and a little bit of a dangerous question. So yeah, I don't know if I can pinpoint one species. What we see is that there is an increase of like the Streptococcus group in the frail individuals. But what makes them, or I don't want to say what makes them more frail, but what we see more often is rather than them having a dominant group, it's just a very unstable microbiota. So there's a lot of shifting. So it's not really that there's this profile that is like in healthier individuals, we see a healthy profile, and if you introduce a bacteria, it's less likely that it will make changes. That's what we don't see in the frail population. So it could be that it's just less diverse rather than one group.
Perfect. Thank you very much.